Hello everyone, I am Neha Kazi from Lilavati Lalji Dayal High School and College of Commerce. In our Science 2 class today, we are going to talk about chapter number 2, Life Processes in Living Organism, part 1. So these are the topics that we'll be discussing in this chapter. So let us begin. So release of energy from assimilated food is called respiration. All right. So because we're talking about an aerobic respiration, glucose is oxidized in three steps, right? So here we first have to understand what respiration is. So it is the release of energy from the assimilated food that is called as respiration. So cellular respiration is done by two methods. Aerobic respiration, that is in the presence of oxygen, and anaerobic respiration, that is in the absence of oxygen. So let us first talk about aerobic respiration. So oxidization of glucose occurs in three steps during aerobic respiration. These are glycolysis, tricarboxylic acid cycle, and electron transfer chain reaction. So let us first talk about glycolysis. So in glycolysis, glucose molecule is oxidized stepwise into two molecules of each of pyruvic acid, ATP, NADH2 and water. So this process takes place in cytoplasm. Pyruvic acid formed during glycolysis is converted into a molecule of acetyl coenzyme A and two molecules each of NADH2 and CO2. Alright, now let's talk about tricarboxylic acid cycle. So when acetyl-CoA molecules enter the mitochondria, the tricarboxylic acid cyclic chain reaction takes place in mitochondria. So acetyl part of acetyl-CoA is completely oxidized, releasing molecules of CO2, H2O, NADH2, FADH2. Alright, so that's about this acidic cycle. Now let's talk about the electron transfer chain reaction. So molecules of NADH2 and FADH2 form during the process, the above process which we saw of tricarboxylic acid cycle. Okay, so they participate in electron transfer chain reaction. Due to this, three molecules of ATP are contained from each NADH2 molecule and two molecules of ATP from each FADH2 molecule. Besides ATP, water molecules are also formed in this reaction. So electron transfer chain reaction is operated in mitochondria only. All right, and your ATP is basically an energy rich molecule. Okay, let's proceed ahead and talk about the energy production on in microorganisms through anaerobic respiration. So basically, if the soil is submerged underwater during germination, seeds perform anaerobic respiration. Similarly, human muscle cells, while performing the exercise, may also switch to anaerobic respiration. This makes the person feel tired due to less amount of release of energy and due to lactic acid accumulation. So some organisms cannot live in the presence of oxygen. So there are many such bacteria that are there. Such living organisms have to perform anaerobic respiration for energy production. Glycolysis and fermentation are two steps of anaerobic respiration. Glucose is incompletely oxidized and less amount of energy is obtained in this type of respiration. Pyruvic acid produced through glycolysis is converted into other organic acids or alcohol with the help of some enzymes in this process. This is called as fermentation. So some higher plants, animals, and aerobic microorganisms also perform anaerobic respiration instead of aerobic respiration if there is depletion in oxygen level in the surrounding. And that example is what I have already given to you all. Okay, so that's about anaerobic. Now let's talk about the cell division that is there. Now in cell division, we are going to discuss two types, mitosis and meiosis. All right. So mitosis occurs in somatic cells and in stem cells, and meiosis occurs in germ cells. So mitosis takes place in two stages, that is karyokinesis division of nucleus and cytokinesis division of cytoplasm. Chirokinesis takes place in four stages. All right, and then it is followed by cytokinesis. Okay, and then we have meiosis, and this takes place in two parts, that is meiosis 1 and meiosis 2. So in meiosis 1 there is crossing over and genetic recombination. Then homologous chromosomes are again divided into two groups forming two haploid cells. So meiosis 1 and meiosis 2 take place in four stages each, which we will discuss further in the slides. All right, then chromosome number is reduced to half in meiosis. Okay, so now let us talk about the prophase. So in prophase, condensation of basically thin bread-like chromosomes starts. Due to this, they become soft and thick, and they start to appear along with their pairs of sister chromatids. So centroids duplicate, and each centroid moves to opposite poles of the cells. Nuclear membrane and nucleus start to disappear. So when it is the metaphase, nuclear membrane completely disappears in this. Chromosomes complete their condensation and become clearly visible along with their sister chromatids. All chromosomes are arranged parallel to equatorial plane, that is the central plane of the cell. Special type of flexible protein fibers, that is spindle fibers, are formed between centromere of each chromosome and both centrioles. Now comes in the anaphase. So in anaphase, centromes split and thereby sister chromatids of each chromosome separate, and they are pulled apart in opposite direction with the help of spindle fibers. So separated sisters chromatids are called as daughter chromosomes. Chromosomes are being chromosome being pulled appear like bunch of bananas. In this way, each set of chromosomes reach at two opposite poles of the cell. And then comes in telophase. So the chromosomes which have reached at top poles, at the opposite poles, all right, of the cell now start to decondense, due to which they again become thread-like thin and invisible. So nuclear membrane is formed around each set of chromosomes reached at poles. Thus, two daughter nuclei are formed in a cell. Nucleolus also appears in each daughter nucleus. Spindle fibers completely disappear. So here you can see what we have just studied about in the prophase, metaphase, then the anaphase, and here finally in the telophase. And also in mitosis, students, it is essential for the growth of the body, necessary for restoration of emaciated body, then for wound healing, for the formation of blood cells also. Okay. Now talking about meiosis. So this is again two stages are there in this, that is meiosis 1 and meiosis 2. So by meiosis, from one diploid cell, four haploid cells are formed. Okay, and meiosis helps to restore and maintain the chromosome number. So let's talk about what meiosis 1 does, and then we'll talk about what is meiosis 2. So in meiosis 1, basically the homologous chromosomes undergo crossing over, and hence there is genetic recombination. So the homologous chromosomes are divided into two groups, and from these two haploid cells are formed. So prophase 1 of meiosis is lengthy phase, which is subdivided into five phases. That is leptotin, zygotin, pachitin, diplotin, and dikinesis. And during pachitin, crossing over takes place. Okay. Then talking about meiosis 2, this is similar to mitotic division. So the two haploid cells that are produced in meiosis 2 now undergo further division, forming four haploid cells. So let us see this in the diagram. See here. So, like I said, the pro phrase is pretty much long in meiosis part 1. So, we have around 5 steps here. Okay. One, 2, 3, 4, 5. See. Then we have the metaphase, anaphase, and telophase. Similarly, how we had studied earlier. So, here we have the prophase, metaphase, anaphase, and telophase. See, so I told you that eventually it will form 4 haploids. Okay. This got further divided and became 4 haploids. So let's talk about Krebs cycle now. So this is also known as citric acid. So Sir Hans Krebs proposed this cycle and hence it is called Krebs cycle. So these are the series of cycle chain reactions which begin with antinucleosine, a molecule which act with the molecules of oxalic acid, and the reactions are cattle with the help of specific enzymes. The first molecule formed in this reaction is called the citric acid. Thus, Krebs cycle is also called as citric acid. So that's all, students. In this chapter, we have quickly done the summary of chapter number 2, and I hope you all have understood this. Thank you so much.